Benim burada olmam.
Good morning. I'd like to thank the organizers for inviting me, and I'd like to thank the president of our university for giving me this opportunity to address you. I'm going to talk about the genotyping studies that we carry out at our university. Well, this is the outline of my speech. Very briefly, I'm going to talk about the Human Genome Project. When we talk about the personalized medicine, it is a project that enables us to think that way. And also in pharmacogenetics, we can understand the disposition to certain diseases through the SNPs. I'm going to talk about the CYPs and our laboratory practices. Of course, the Human Genome Project is one of the biggest projects that takes us to this day. The Human Genome Project was started in 1990, and it was completed in 2003. And this is the highest budget project covering the biology field, and it was started by the US, by the relevant ministry, and it was actually started in 1960s because they tried to understand and investigate how the radiation would affect the genes of astronauts. And then it was widened in scope, not only the astronauts, not only the radiation, but also the other aspects, the other effects on the DNA were started to be investigated, and the Human Genome Project started in 1990. In fact, it is not concluded yet. It is still ongoing. What was the purpose of this project? One of the aims was to identify 30,000 genes in human DNA. And at the beginning, we were thinking about the millions of genes in the human DNA, but at the beginning of the project, they were talking about 90,000 or 100,000 genes. However, this project has shown us that the genes are much fewer than we expected in number, but their structure is much more complicated. This project was not only about understanding the number of the genes, but also they tried to understand the structure, composition of the genes, and of course they tried to have a database for this. And at those days they were trying to have a patent for this investigation. And right now we can have this information shared with the public. Another aim was to have bioinformatics. Yes, we do have the information data at hand, but how can we use that information? And of course, as the ELSI, the the ethical, legal, social issues were also addressed in this project in order not to cause any problems. Until the beginning of 1997, the project was going very slowly. At those times, actually, the technical discussions, debates were taking place. But in 2001, the first draft was declared, and in 2003, the final results were declared. So, very briefly, what does it say to us? Right now, we have a great amount of information about the genes. We know about our number of genes, and we say that 99.9% of the individuals have the same sequence. Actually, the nucleotide bases are exactly the same. And we only have the differences in that 1 per 1,000 part of this sequence. And also, we can also guess the longer gene sequences. And we know the number of the genes on the chromosomes. And also, approximately in 1.8% of the genomes are coding proteins. And the remaining part is not very well known in terms of function, but we can say that they are taking part in the genome metabolism, but we don't know what they do. and 50% of the sequence is repetitive sequences. First, they were named as junk DNA, as they did not do anything. But right now, we know that the lymphocytes are changing their genomic structure very easily, and we know that this junk DNA has active role in that process. One of the most important aspects is the variations. This is the 1 per 1,000 area that we have the differences, and these are the SNPs, the SNPs, as we call them. For instance, although we may eat the same food, we may get the same treatment, but people react to these treatments or nutrients differently. For instance, one person may eat the same as the other one, but he can get hypertension or some other diseases, while the other person does not. So what are these SNPs? For instance, this is a DNA sequence, and normally this is the highest frequency in society. This is called an allele. And if there is a variation in this system, and when you look at the rate in the society, and if it's more than 1% in the society, then we call it as SNP, the SNP. What is mutation? If the variation is less than 1% in the society, then we call it as mutation. So where can we see these SNPs? Of course, it can be inside the genetic area, genetic site, and it can cause the RNAs, form the RNAs by means of mutations or variations. And in the regulating areas of the genes, they may cause some differences, and they may cause problems in the regulation of the genes, or they may not cause any problems, and they can be also present in the areas where there is no outside the genetic area. And when we are doing the haplotyping, then these SNPs are taken as bases, and therefore we can understand the categorization of the haplotypes. In 2000, NIH, the National Institute of Health, started a project. This was a 3-year project, and it was called HapMap project. 3 million SNPs in human were trying to be identified, and they tried to understand their connection with certain diseases. So how are these activities going on? Right now, the SNPs can be identified very easily. Once they are defined, of course, their areas are important. Are they in the coding areas or non-coding areas? If they are in the non-coding areas, then we can make the haplotype analysis. So we can have some small connections, and we can understand the predisposition to certain diseases. Or if they are at the coding areas, then again we can carry out those kinds of analysis, and haplotype groups can be formed, and we can understand what kind of diseases that person may have. And also genome-wide association studies are carried out in order to understand the localization of the SNPs. Of course, there are still many things that we don't know about the Human Genome Project. The studies are underway, they are still going on, and there are so many projects about this topic, and we are not knowledgeable about the full functioning of the genes. But what is the most interesting part for us? It is the pharmacogenomics and maybe the gene therapies that we may have in the future. The aim is very easy. We are trying to predict the diseases, and we would like to have more realistic and tailor-made treatments for people. The previous speaker talked about this CYP1 and CYP2B6. These are present in 17 different families in 57 different genes, and we know that there are some variations and there are polymorphisms. On the basis of this information, we can understand the enzymes' functions, whether they are metabolites or they are having some other functions. And in the gene areas, for instance, these vertical lines are the variations, and we can see the differences between the people who are responding to treatment and who are not responding to a treatment. So we can understand the areas where the differences are between those two individuals, and we can understand their enzyme systems, or through CYP2D6. Here you can see the chromosomes of the genes of CYP2D8 and CYP2D7. We think that these chromosomes can derive from certain genes, or they can be active during the embryonic period. CYP2D6 have 9 coding areas, and in between it has some non-coding areas. You can see the synapse here, the SNPs here, and when we analyze them, we can categorize the haplotype groups. We can also show you the most frequent mutations, and next to them, I mean around them, there are so many SNPs. And in genotype and phenotyping, we do not have the full matching components, but maybe these SNPs are causing these variations in this wide area. Again, here you can see according to the alleles and haplotypes. For instance, here we have the haplotype, and we can look at the alleles in that haplotype, and we can say whether it's poor metabolizer or which haplotype it belongs to. We can understand this through this analysis. And once we understand that, what do we do? For instance, we have the codeine metabolism. If we cannot find enough activity through the CYP2D6 analysis, we can go for alternative treatments. Or when we look at the dopamine receptors, there are different areas that are involved in this process, and according to its response to dopamine, the differences may occur in response. Therefore, in individuals, we can have some different and variable responses to the same treatment. So, how can genetics be useful for us? Well, we can understand the optimum dose for a person, and also we can save some time, and we can get rid of the side effects, and the clinicians can also be protected from very challenging situations. Of course, the medical costs are reduced as a result of the genetic analysis and tailor-made treatments, and also FDA is also focusing on these genomic studies. In many CYP2 including systems, the FDA is ready to give the approval. What do we do in laboratory? Once we isolate the DNA, we do the genotyping. There are some different methods. The most frequently used are the direct sequencing and fragment analysis. But of course, in bigger centers, the next generation sequencing or array-based chip technologies are used as well. In the first samples that we receive, we can multiply the areas that we would like to have. We can use the gel, and sometimes we can use the NLP implementations, but we can have the Sanger sequencing, and we can look for the mutations in areas that we would like to see. Or if we would like to identify the problems in the areas that are predetermined, then we can have the real-time applications. For CYP2D6, the number of copies is very important. If there is no match in the sample that we receive, then we use a superior technique, which is the comparative hybridization method. Therefore, we can have the analysis of the number of the genes. This is a comparative table, but I don't want to take much of your time. But all the techniques are open for development. These are validated techniques, but of course the implementation and cost can differ according to the places that you implement this. What happens once we understand, make the analysis? We can understand the haplotype of the individual, and we can understand the enzyme and genetic systems of those alleles. And also for CYP2D6, this is the example that I have shown, and it is the same for CYP2C9. You can understand the haplotype, and you can understand the variations and the variation sites, and we can understand some mechanisms about the enzymes. And this is a chip sold quite intensely by Roche. and this chip can identify 26 digits of these categorizations. And I'd like to, of course, I would like to thank all my colleagues and my professors who spent efforts.